Okay, good afternoon and welcome to our final health talk of the day. My name is David Obershaw and I'm on the Stanford Medicine Community Council. I'm also an adjunct lecturer in the Department of Electrical Engineering here. Before we get started, I want to remind you, here's the prop for this. There's a QR code in this thing. If you want to capture the information right there, you can hop on. And for people that are watching from online, the code is also available online. We'll have a live QA, and using the QR code, you can input your questions. And we'll be able to field those at the end of the presentation. And now I'm happy to introduce Dr. Karl Deisseroth, a practicing psychiatrist and leading neuroscientist and researcher here at Stanford. He's also the D.H. Chen Professor and a professor of bioengineering, psychiatry, and behavioral sciences. And on a personal note, I would add he's a terrific writer. I just read his book, Projections, and he beautifully balances, the frontiers of neuroscience with our distinct humanity. It's a wonderful book, and I'm not getting commissioned on it. It's just a wonderful book. You got to look at it. All right, today Dr. Deisseroth will speak about his research that explores the origins of our emotions such as fear, sadness, and happiness. As you'll soon hear, Dr. Deisseroth is exploring how our feelings are causally connected to activity happening in specific regions of the brain and how disruptions in circuit systems can lead to conditions such as depression and anxiety. This groundbreaking research, which you're to hear about first, has far-reaching implications for our understanding of how mental health conditions emerge and therefore how treatments can become more targeted and precise in the future. And now, please join me in welcoming Dr. Deisseroth to Health Matters.
Thank you. All right. Thank you, David. Can everybody hear me okay? I'm really delighted to have the chance to speak to the community and to share some of the excitement of what's been going on at Stanford and where this will lead us in the future. I'm a psychiatrist here. I've been at Stanford for a while, 30 years or more, actually. I came here as an MD PhD student back in the 90s. But we've actually made a lot of exciting progress very, very recently, and a lot of the basic science research, I do most of my basic science research in the building right behind you, the Clark Center. It's a building where we have animals, mice, and where we work with genes and fiber optics. But very recently, I've been able to use all what we've developed and bring things to the hospital, to Stanford's beautiful new adult hospital, actually both the adult and the pediatric hospital, Packard and Stanford both. And some of our new research rooms, I'll tell you what we're doing in these rooms are shown here. So it's an incredibly exciting time, and a lot of this, even just in the last few weeks, is coming to fruition, and I'm excited to share that with you. But first, going back in time a little bit, so I mentioned 30 years ago, this was me 30 years ago. I cut my own hair, so that's why it's long in the back. It wasn't intentionally a mullet, but I just cut what I could see in the mirror, and I couldn't see the back, so. But what I'm doing there, I was again, just a few feet from here, I was studying how neurons work, and we were using single electrodes, poking single cells with them in Petri dishes in the lab. So very precise, but single cells. And flash forward now, 30 years later, I still don't cut my hair very well. But now, electrophysiology of human brains, people who were guiding experiments and insights from the animal work, from that very basic work that we've been building toward for decades. And just now, things are really starting to come together. So what is the essence of the technology? What is the essence of the insights? And I like to see how things are connected across time. You saw 30 years there. I like this quote from T.S. Eliot. He says, "We shall not cease from exploration, and the end of all our exploring will be to arrive where we started and know the place for the first time." And this is true in science and medicine. It's also true in biology. A lot of the tools that we are using come from ancient molecules and ancient structures. Billions of years ago, before there was multicellular life, the oceans were full of single-celled organisms, single cells, and they got energy from light, not with photosynthesis, the chlorophylls you see at the lower left, but from the much simpler structure you see at the right. This is a molecule like vitamin A. It's called a retinoid. And light was harvested for energy with a light-activated generator of electricity, a little protein that received a green photon and moved a charged particle, an ion, across the membrane of the cell. If you move a charged particle, that's current, that's electricity. And so these cells, filling the oceans billions of years ago, made electricity from light. What we've been able to do at Stanford is to use this ancient approach because the brain's electrical. And so we had the thought more than 20 years ago to start bringing these ancient proteins, billions of years old, and put them into brains and turn light into electricity and control neurons, control brain cells with light, turning them on or off. And this is something that we call optogenetics. And over the years, many thousands of papers all over the world have been published using this methodology, but I'll give you just one example that's very recent. It's been applied actually all across the body. You can put these light-activated proteins in any cell type in the brain. You could put it in dopamine cells or serotonin cells, excitatory or inhibitory cells, turn them up or down with a flash of light, with a turn of a switch, and you could see what happens to behavior, to cognition, to memory, to motivation. We can turn up or down the ability of an animal, starting with a mouse, but now we're getting into human beings. We can turn up or down any kind of motivation or function or priority of an animal. And this is a way of understanding the causal role of cells in complex brain function. But we can also extend to the heart. And this has been very exciting. There was a very ancient question. What is an emotion? What is a feeling? And psychologists and philosophers have pondered this question for a long time. Psychiatrists like myself would like to know this. I'm sure all of us would like to know this. But it's a very hard question. William James, the American psychologist more than 100 years ago, thought that an emotion required the body. It wasn't just in the brain. He said if you took away all the physical bodily manifestations of an emotion, whatever was left wouldn't be an emotion after all. And he thought about the pounding of the heart and the higher respiratory rate in fear and anxiety. He said if you take those away, you don't have the emotion anymore. Very interesting perspective, but very hard to prove, very hard to test, because you'd have to provide experimentally that elevated heart rate very purely and see what happened. And that's very hard to do. The heart is a big organ. It's moving a lot. It's got a lot of cell types in it. It's got a lot of neurons in and around it. It's got a lot of blood supply. Very hard to pace the heart precisely. Well, with optogenetics, actually, we could do that. We used a special kind of genetic targeting, and what's seen in the lower left is a mouse heart, and it's got that opening in the middle where the blood goes through, and that sort of magenta coloration around the rim, that's us putting in one of these ancient light-activated proteins, these billions of years old proteins, and now we can pace the heart precisely with light. We can drive muscle contraction in the heart at any rate we want. And we put a little LED vest around the animal, very fashionable little bit of clothing to pace the heart. We have to generate the light somehow. These molecules are so sensitive that we don't need surgery. The light gets through the skin and gets to the heart. And we make it safe so it doesn't elevate temperature. And in the lower right, we can pace the heart rate at any level we want. Mice have hearts that go at 600 beats per minute normally, but if we want 700, we can do it, 800, 900. And we found indeed that if you provide an elevated heart rate to a mouse, it has a very interesting and specific elevated anxiety when it's in a risky situation. And so this ancient question from William James and going much farther back turns out to be right, that you need the body, you need the manifestations in the body for the full expression of emotion to be felt. So that's one very recent example. A great team of postdocs and grad students helped me get to that answer. We can control a big organ like the heart. We can also go down to single cells, and we use holograms. What's shown in the lower panel is what's called a spatial light modulator. It makes a hologram, and it projects in spots of light, single cell-sized spots of light. And in the top row, every little red circle is around a single cell in visual cortex, the part of the brain that controls vision in a mouse. And we've picked out, those little red circles you see is us, we've picked out those single cells that we want to stimulate, and then we're using the hologram to play in spots of light to hundreds or even thousands of individually specified cells. We can turn them on or off at will in a freely moving animal. So it's not just an organ, not just a cell type. We can really play even better than a conductor of an orchestra could do. We can get down to every single musician, if you will, every single cell, and turn them up or down at will and see how cognition, action, behavior, perception, motivation arise from the activity of individual cells. So this has been a research tool. Thousands of papers have been published using it, sorting out the causal role of cells in all kinds of function. But you might ask, how is this going to affect medicine? How is this going to affect psychiatry, neurology, neurosurgery, or cardiology, or beyond? And you can imagine a few paths for this, and these are working. So one thing at the top you might think of as direct translation. That's where we'd put these ancient proteins right into people. And we can do this. And this has been done. So you can actually put these light-activated proteins, one of them is called a channel rhodopsin, and you can put it into a virus, a safe, well-tolerated virus, and you can put that into the central nervous system of a human being. And this goes beyond the basic science, which is still going on very strongly, and exploration of nature, exploration of how animal brains work has been incredibly powerful. But my friends in Switzerland, you heard Switzerland mentioned earlier for its cleanliness, they're also pretty good at translational neuroscience, and we gave them a lot of our clones over more than 10 years, and one of these projects led to a blindness cure in human beings. They targeted retinitis pigmentosa, which is a neurodegenerative disease of the retina in human beings, and they put these light-activated proteins into the retina of a person who was completely blind, conferring new light sensitivity onto the cells that were there, and this blind person could now see and reach for objects on the table that he couldn't see before. So this works, direct translation works, but even bigger opportunities, what you might see in the lower panel here, indirect methods. If you know which cells cause anxiety, if you know which cells cause reduced motivation or hopelessness, or the inability to enjoy things, or conversely, what we see in psychiatry, sometimes over enjoyment of things. People who have bipolar disorder or mania can be over reward driven. If we know the cells that cause these things, then we could design medications to target those cells. And so it doesn't all have to be a direct virus protein into a human being. It's understanding that's the most important thing. So this approach is also starting to work, and across all the domains of psychiatry. David mentioned the book I wrote. If you're interested, you feel free to check it out. But each chapter comes from a different patient that I've seen right here at the Stanford emergency room. Acute psychiatry, people coming in, lives shattered in the very depths of the most acute psychiatric illness. And each chapter is a different condition. The first one is depression due to a bereavement. The next one's mania. Then there's autism. Then there's borderline personality disorder. Then there's schizophrenia. Then eating disorders. Then dementia at end of life. And in each one of these, I start with the patient's story and then share a little bit about the evolution of humanity, why we have these conditions, and then a little bit about the optogenetics, how this optogenetics is helping us understand these disorders and to treat them. How is this starting to extend into modern clinical medicine right at Stanford? Well, I'll tell you how that's going. And this is the genesis of a new program at Stanford that's been supported very powerfully up and down the Stanford community, from the president and the provost to the heads of the hospitals, the dean of the School of Medicine, and everybody who works with them. And we got down this path by considering a very thorny and mysterious and very interesting problem in psychiatry, which is called dissociation. This is a painting by the American pop artist Andy Warhol, and you can see he's fragmented. His perception of himself is separated into at least 3 parts. This happens in life. Dissociation is a state where you're still conscious, but parts of you are broken apart, separated. And this could be different parts. It might be, you might no longer be able to feel emotion about something, a sensory stimulus. You might be perfectly aware of damage being done to the body, but you don't care about it. That's one kind of dissociation. Or you can have an out-of-body experience. You can really feel that your body is not your own. Now, these are very interesting and complicated things, and a lot of people, if you haven't experienced them, these symptoms, it seems a little funny, right? How could that really be happening? But it's actually pretty common. Dissociation can happen in PTSD. It can happen in dissociative disorders, borderline personality. Drugs like ketamine or PCP can cause these. Trauma, 70% of people who experience trauma will experience dissociation. So I can guarantee, unfortunately, many people right in the audience have dissociated. And it can be a scary thing. It might be part of how we're designed. It might be part of separating out the emotion about sensation from the sensation itself to allow people to survive in very challenging moments. It's a very real thing, though, and very interesting. How does this happen? And when it gets too extreme, like most of psychiatry, it can become a real problem. What's interesting is the commonality of the descriptions people use. Someone with a dissociative disorder will say, if my mind is a car, I'm in the passenger seat, looking at myself driving. Someone on ketamine would say something very similar. You're in the audience. You could watch the movie of your life and judge every aspect of it without any sort of emotional reaction. That's what it's like. Different causes, same symptom. And this is one of the things that really helps us as a scientist, the repeatability of it, to study it and see what causes it. So we thought, well, let's start with animals. Let's see what we can see. Let's give ketamine to mice and see if we can make them dissociate. And we built some complex optics to let us see all across the brain in these mice. And we compared if they were just given a salt solution or ketamine. And in the ketamine case, what you're seeing is brain activity all across the brain of a mouse. And in the ketamine case, you might see that red flashing thing in the middle bottom. There was a part of the brain called the retrosplenial cortex, RSP, that started to pulsate with ketamine. And it was not connected to anything around it. It was doing its own thing. This was never seen before in any other condition, like with the salt solution or saline solution. This was a dissociative drug causing one part of the brain, a big part of it, to do its own thing and to not be connected to what was going on around it. So we were seeing, we thought, dissociation right before our eyes, a separation of one part of the brain from the other parts of the brain. So we then decided to explore this in human beings. All right, now how are we going to do this? Well, we're not going to give PCP to people, but ketamine, as it turns out, yes, it does cause dissociation, but it's a safe, approved medication. At higher doses, it's used as an anesthetic. At lower doses, it's an FDA-approved antidepressant. If you read the package label for ketamine, it says one side effect you may get is dissociation. So with our team, one thing we've done in this new program, the Human Neural Circuitry program we started at Stanford, we took advantage of the beautiful new hospital and the epilepsy monitoring unit. And this is where people who have intractable epilepsy come and spend a week or so in the hospital with recording electrodes placed all around their brain, including deep, so the neurosurgical placed electrodes, 10 to 15 of them all around their brain. And they wait for a week in the hospital to have seizures. They want to have seizures because that lets the neurosurgeons know which part of the brain needs to be modified to reduce the seizure incidence. But this is an amazing opportunity. We have people sitting there in the hospital, not doing anything for a week, waiting for seizures and electrodes all over the brain. And we thought, okay, in a very careful, safe way, let's go and give a single dose of ketamine to these people and see what happens with the recording. And so we did that, and we said, let's also do this same experiment in mice at the same time, and let's look for conserved things. Humans and mice diverged from a common ancestor 70 million years ago. If something is conserved between these two species, you better believe it's important, and that's going to be a big clue that it's a significant thing to study. And so that's what we did in humans and mice. We gave ketamine to both. We recorded across the brain in both. We gave little brief sensory stimuli like a little puff of air, and we used all kinds of careful clinical tests with the people to see what they were experiencing. And what we found is really striking. First of all, what's shown in the left is every little spot is a spot in the brain where we recorded from. You can see we got pretty good coverage across the whole human brain. And in the bottom trace, you can see each column is a different participant, a different patient, a volunteer for the study who gave their consent to have this procedure done. And the numbers you see there are how dissociated they became. And some people didn't dissociate very much. That one in the middle with the 1, 0, and 0, that person didn't dissociate much with ketamine. But most of the others did, and we quantified that. And then we could look and see which part of the brain is changing in this human being who's telling us with their own words that they're dissociating. So getting to the physical, cellular basis of this very complex and mysterious symptom. And we were able to address this. And what we found is illustrated in the lower 2 rows here. What we found is that normally, if you give repeated puffs of air to someone's eye, like you might have as an ophthalmologist, it's not painful, but it's a little annoying, and after a while you'll start to become sort of bummed out about it. You'll have a negative emotion about it. Not painful, but we asked the people, how are you feeling? And they said, I really don't like that. I'm feeling kind of mad about it. And all consented for this protocol, by the way, fully informed. But then when we gave them ketamine, they were fully aware that it was happening, but didn't care. The same person, minutes later, we continued to give the air puffs, and they were just eyes wide open, waiting for the next puff. And we talked to them. We said, "What's different?" "Oh, it's kind of interesting. It's fine. I don't mind it." And then when the ketamine wore off, they went right back to that negative emotion state. So we could turn it on or off at will. And we found the spots across the brain that indeed were dissociating, separating out the activity in this human brain, one part separated really physically from another. So with our own eyes, we can see, not just in mice, but in human beings, what dissociation really is. So this has been incredibly exciting. We're now extending this to non-invasive measures. So you might imagine not everybody's going to have an electrode placed deep in their brain. But it turns out, as you can see in this picture, we're now picking up the same signal with surface scalp EEGs, non-invasively. And it's harder to see, but now that we know what to look for, based on our earlier work, we can home right in on it and measure it with a non-invasive measure. And this work is going to be published within less than 2 weeks in a major journal. I'm not allowed to say which one, but if you look in a couple weeks, you'll see it coming right out of Stanford. And I want to say this is an amazing team effort. The prior talk, you and Ashley talked about how amazing his team was. I'm so grateful to be here at Stanford and have the students, the postdoctoral fellows, and the colleagues, the neurologists, the neurosurgeons, the nurses, the patient care managers, the EEG technicians. I'm quite sure this could not have been done anywhere else. The nature of the community and the openness and the collaborativeness is absolutely mind-boggling here. It's been hard. Not saying it hasn't been hard. It took about 5 years to get this work done. And the late night phone calls, texts from patients or nurses or attendings worried about one thing or another. It's been a challenge, but for me, this is what we're here for, and it's been incredibly rewarding to get to this point. There's so much that comes from the future now, too. So I'm a psychiatrist. I study what I've told you about, but the people I treat, I'm particularly interested in autism. This is the patient population I spend most of my time with, along with folks with depression. And what we can now do is apply this same principle, everything I just told you. Now we're going to look at autism, and we're doing this in the pediatric hospital, Lucille Packard Children's Hospital. And a lot of kids come there with electrodes placed for epilepsy as well, and about half of them have autism. Autism and epilepsy go together quite significantly. They're comorbid, as we say. About half of the epilepsy kids who come for this by chance also have autism. And that's an amazing opportunity. No doubt poses challenges, but it's an amazing opportunity because now we can run the same playbook and see what really is autism. What is actually physically is autism. And so what's the equivalent of the FAST test that we're doing? Well, one thing we're doing is looking at eye contact. And you might know that folks with autism have a real challenge with forming and maintaining socially appropriate eye contact. It's a very fast behavior. The movements of eyes are as fast as the air puff or even faster. And we can sync that up with the EEG recording. And the movie I'll play here is an example of a recording from a child at Packard Hospital. Again, we have all the spots, we can see all the activity, and we play the movies, age-appropriate movies. It might be a kid's thing for for someone under 10. This is a clip from Parks and Recreation for a teenager, and you can actually see we're tracking the eye position. The red dot in the upper panel shows exactly where the child is looking. When are they making socially appropriate eye contact, and when are they looking away, as they are here? And then back to the eye, look at the mouth, which a lot of kids with autism do. They focus on the mouth instead of the eye. We can look at pupil diameter, but everything exactly synced to what's going on in the brain. And so this is, for us, an incredibly thrilling opportunity to get to the physical nature of this very challenging, in many cases, condition. Autism is one example. Everything I mentioned is also available for deeper exploration. Everything from depression to eating disorders to personality disorders, schizophrenia, mania, and dementias. So I'll wrap up there. I'll just say this is, for us, a very, very special moment. Again, it couldn't have been done anywhere else, and the future is very bright. I'm so grateful to everybody who's worked with me, and grateful to all of you for taking time on this beautiful day to come and learn about it, and happy to talk and answer questions. So thank you.
Thank you. Thanks, Dr. Deisseroth. Okay, thank you so much, Dr. Deisseroth. We're now going to, for sharing all this information, and I think there's probably a lot of questions. And so, again, I would remind you, if you want to input a question, use the QR code. But we've got a variety of questions ready to go here. So, all right, let's start with, let's pull up a seat. How close are we to developing treatments for mental health conditions based on the work related to emotions you've been doing?
Yeah, so this is the key question. When does the insight into the physical nature of the illness actually lead to a treatment? For cells and structures we understand, like the retina, we're already there. And so we can take someone who's blind and we can make them see again. For the psychiatric diseases, we're still sorting out which cells are actually causal in the symptoms we're talking about. And we've made a big leap forward with dissociation. We're making big steps with autism and depression. And then that indirect path that I mentioned is probably the fastest. It's now that you know the cells that matter, build medications that target those cells. And there are a lot of ways to do that, including with genomics and other strategies. And that's already starting to happen. There are companies working on this route of building on that causal knowledge and creating medications. So I think within a few years, you'll start to see medications start to become tested and available.
Excellent. Okay, are there specific emotions that have proven particularly challenging to study?
Yeah, this is one of the things that's most challenging is, could an animal express that emotion? And in cases where an animal, like a mouse, seems to express something like that emotion, the path is very clear. And that's true, actually, for a surprisingly large number of psychiatric symptoms, but not all. And so the most challenging ones are those that hit these most complex integrative functions of the human brain. Schizophrenia is very hard. There are not good animal models of schizophrenia. A lot of what's challenging in schizophrenia is very high-level things that only human beings do well. People have what's called a thought disorder in schizophrenia. They can't organize their thoughts in a coherent pattern and make a plan that makes sense and keep track of complex conversations. And it's very hard to study those things in mice where they don't have that capability anyway, as far as we know. But there's hope. You know, if we look at what's happened in dissociation, few would have thought that we could make headway on that, and that was a very tricky, challenging one. Autism symptoms, you know, mice don't make eye contact, but they have other very fast social behaviors, and so we're having the same sort of translational opportunity even for autism. So most of these symptoms actually are surprisingly accessible.
That's great. Has your team's funding been affected under the new government policies?
Yeah, so this is a big issue. There's a lot of very justified anxiety and stress across the country in the scientific community. And I haven't had any, personally, I haven't had a grant canceled or taken away, but we think that might happen in the future. One thing that's been fantastic about being at Stanford is, in addition to NIH funding, there's a very supportive network of donors, community folks who've pitched together and supported a lot of the labs, and that's been absolutely crucial for us. All of the human neural circuitry program work I described to you, we would not have been able to get there without donations from the community.
Well, that's great.
So we're surviving
Yeah.
for the moment.
Well, fingers crossed. How does ketamine causing dissociation help with depression?
This is a big question. I talked about several things that ketamine does. One is it's an anesthetic. At high doses, anesthesiologists use it really to reduce the consciousness level. But at low doses, it's an antidepressant. And then it has this dissociative side effect. And a big question has been, are those last two bits related? Is the dissociation, which is framed as a side effect, what if that's actually part of how it generates the antidepressant effect? What if this separation off of emotion in stress is maybe part of what's going on as an antidepressant? So I will say it's actually not known that the dissociative effect is causal in depression. That's an area of some controversy in the field. I would say most people actually think they're separate, but to be determined. I think it's an extremely interesting question, and I do think dissociation is an adaptive response. I think it's part of helping mammals continue to function, continue to cope, even in life-threatening or very challenging situations. And I think that's why the capability to dissociate has been maintained between mouse and human.
Yeah. All right, I mentioned how emotions interact with the body and our physiology. I noticed studies finding increased dementia risk in long-term unhappy marriages, for example, and wish to understand how this may happen. Very personal question there, yeah. Any divorce lawyers? No. Yeah.
Actually, this association between dementia and depression symptoms is known, and that's been studied, and actually in the last chapter of Projections, I write about that quite a bit, actually. It's not clear which direction the causality goes, actually. They definitely co-occur, worsening depression and worsening dementia symptoms. But you could imagine various arrows going both ways, right? People who are depressed very often have low energy, low exercise, poor diet, elevated stress levels, and that certainly can affect dementia. It can put you at greater risk for strokes, for microvascular occlusions. So that's certainly the case. But then, of course, people with dementia will have reduced ability to engage in helpful interactions with their spouse, and then there'll be a breakdown in the dyad of communication that happens. So I will say it's not known exactly how that happens. I do think that what's most clear is that dementia certainly can cause depression-related symptoms. That's very clear. People immediately after a stroke or immediately after a series of transient ischemic attacks, they can end up with depression symptoms quite clearly, and that can disrupt all of their social functioning, including marriage.
Sure. Yeah, makes sense. Okay, can your work be applied to people with ADHD?
Yes, ADHD is a really, really interesting and important cluster of symptoms, and very relevant. All the folks with kids may, will have wondered now and then, does my kid have ADHD? Because the symptoms of ADHD are expressed by every single child that has ever existed. There's no question about it. But like much of psychiatry, it's a level, it's a spectrum, and at what point does it become hard for that person to function in society? And that's really how we define disorders in psychiatry. We don't make value judgments. We don't say this is bad, this is good. At the end of every psychiatric diagnosis process, there's a question: is it disrupting this person's social or occupational functioning? If the answer is no, it's not a disorder.
Right.
We just accept that. It's not a disorder. And that I think is helpful. ADHD, though, to what extent is it representing an energetic state, a novelty-seeking state that is positive in some sense, that is valid, that is helpful, that in our evolution as the human family has been important for us to get to where we are. Nevertheless, it can cause challenges in our current society, and we want to help people if we can. Now, ADHD, we don't have right now great animal models, but there's a lot of interest in it, and particularly autism. What's one of the key distinctions that we have to make diagnostically? Does a child who's having trouble in school, trouble with social interaction, is it autism or ADHD? So we end up studying both anyway,
Yeah.
and kind of one serves as a control for the other. So we may get ADHD insights while studying autism.
That'd be great. All right, dates of depression are increasing worldwide. Is this a nature or nurture thing? If someone has treatment-resistant depression, is it likely caused by genetics?
Well, first of all, like you and Ashley said right before, it's amazing how many disorders are about half nature, about half nurture, and sometimes it's 25-75, sometimes it's 60-40. It's definitely the case for depression. It's got a significant genetic risk, no question. Monozygotic twins who are raised apart, if one has depression, the other is more likely to have depression than somebody from the general population. So there's no question there's a genetic link, and many genes have been identified that contribute. However, there also certainly is a nurture thing. People who have elevated untreated anxiety for a year or more, that's got a risk, about a 50% risk of translating to depression. And so people who've had elevated, unmanaged anxiety due to life, due to situations, they'll have a risk of progressing to depression. Now, as far as epidemiology increasing, of course, the world's changing. The world's always stressful, maybe more stressful now than recently. But honestly, people have always said that. There have been a lot of bad times in human history. I think the real issue is when we see rates go up, like for autism, like for depression, a lot of it is increased understanding, increased identification, increased diagnosis, which is a good thing because we're identifying people, we're correctly saying, hey, here's somebody who has this cluster of symptoms that may be due to this, and maybe we can help them this way. Now, treatment-resistant depression also has some genetic correlation separate from depression itself. And so I would say if people have treatment-resistant depression, it may be valuable, not certain, but it may be valuable in some cases to do some genome sequencing and seeing if there is a mutation or an allelic variation that may help improve understanding. It doesn't lead to treatment decisions right now, unfortunately. That could be a nice intersection between the kind of work that I've described here and the genomic revolution, is that could lead to us finally being able to do something about those genetic links.
Yeah, that'd be great. Can your research help explain why some people are more emotionally resilient than others?
Yes. Now, this is one of those things that we can study in animals very well. This is the diversity in resilience.
There's tough mice.
Yes, there absolutely are, and they can be siblings. They grew up together, and one of them's and almost have the same genome, but one of them is resilient, and one of them is not. It's amazing, and it's consistent. And so we can actually study this very, very clearly in mice and identify patterns that seem to promote resilience. We have not yet translated that to the human case yet, but the path is quite clear for that. We have good animal models and a lot of things you can see correlates of in various kinds of tests. We can give, without causing too much damage or stress to people, we can put them through sort of games that cause disappointment. Mm-hmm. Or positive feelings, and we can see who's more resilient to the losses, to the disappointment in these games, and see what's going on in the brain as these things are happening. And so that's something that we're excited about exploring.
Yeah, very cool. Do you have experience or thoughts on EMDR in the treatment of trauma?
Yeah, for folks who don't know, EMDR is an eye movement behavioral therapy. So people who have trauma of various kinds, They'll go through sessions of eye movement guided by a therapist, following a finger, looking back and forth for a prolonged period of time, many minutes. And there is some data suggesting that can help some people with certain kinds of trauma or personality disorders. No one's ever quite known how or why that works. I've never carried that out myself, so I can't speak to whether it's actually efficacious or not. But people do, many people do believe it helps. The rhythm is interesting. It's often a few times a second, and that has an interesting parallel with that sort of rhythmic flashing. I showed you earlier, it was about 3 times a second, that ketamine-induced dissociation rhythmicity in that little patch of the brain. And so it's theoretically possible that this EMDR could be causing a part of the brain that's tracking this hand movement and caused by this eye movement to oscillate out of sync with other parts of the brain, and might cause a dissociative-like effect, but unclear at the moment.
Okay. All right, is there an impact on human emotion from social media, screen time, and technology? What about social media addiction?
Yeah. This probably is relevant too. I will say I think more research needs to be done on these questions. It's very hard to do controlled experiments with this sort of question. So we can't prospectively define 2 groups of people that are matched in various ways and say, okay, you're going to scroll through your phone a lot and you're not going to. That would be great, but we can't do that. So we don't really know. Definitely, there are people who are on social media to extreme levels, certainly do have increased levels of mood disorders. Cause and effect, hard to distinguish there a little bit, right? And addictive properties of social media do seem to be real as well. Why do these extreme cases happen where people are really on their phones all day and all night? And there is a reward process that happens with social media that can tap into the same circuitry that drives other kinds of motivated behaviors. So I think that's real. I think that is likely to have some properties of addiction. It's not true addiction in the sense that the body does not become physically dependent, as we think about for drug addiction. That's the real problem with drug addiction, is the physical dependence of the body. But the cognitive dependence, the emotional dependence certainly can become disruptive, and so it fits some of the criteria of addiction. Mm-hmm. That's the kind of thing that we could make headway in as well, and we're hoping to explore these sorts of things. It's very well adapted to, again, the kind of inpatient setting that I talked about. You've got a week where you can work with people, you can let them have the social media pattern that they're interested in, or you could ask another group to not do that. And you could study the brain activity patterns that were present in those 2 groups.
So cool. Can your work help us understand the neurological basis for empathy?
Yeah. Empathy is such an important property that human beings have and can express, and I think very essential to our survival as a species, as the human family. We would like to understand it. Some people have less empathy than others. That's very clear. There's a disorder in which people have essentially no empathy. We call this antisocial personality disorder. It's related to high rates of criminality. And this does have a genetic link, so it does run in families, and it extends across the life. So a lot of people who have adult criminality and display a lack of empathy, they showed those traits very early on. And when we're carrying out the diagnosis, we'll ask about childhood. We'll say, are there, were you, we'll talk to the family, we'll talk to the patient, was there, harming animals as part of your childhood and things like that. And a lot of these things, they do show up as lifelong patterns. So genetic link, lifelong patterns. And so if it's possible, this helps me as a doctor feel some empathy for those people lacking empathy, because they were never able to manifest it due to something about their genetics or the structure of their brain. It's hard, but but we can understand it and frame it as a disorder of people who need help. So the fact that some people have it, some people don't, that it's a stable, lifelong pattern does open the door to studying it. It's somewhat difficult to study, I would say, in animals. So I think this would really have to be a human study. There are people who claim mice or rats can show empathy in various ways. Probably to some extent, but very hard to separate from kinship relationships and other things that are less interesting. But with human beings, we could make progress on that, again, with the sort of framework that I shared earlier.
Very cool. All right, this is the last question. We're hearing so much about AI in medicine. Is there an application for it with your work?
Well, we use AI quite a bit. Not in a dedicated way. We're not an AI lab, but we find it useful in a number of ways for helping us sort through data. A lot of our our eye tracking software that I showed you, and tracking the blinks and so on. There are AI tools that have greatly accelerated that work. And so yes, short answer is yes. I'm also a teacher here. I teach the undergrad, I run the undergrad bioengineering program here at Stanford, and I will say AI has really challenged our teaching.
Given their ability to generate good output?
Yeah. So that is very much an unsolved problem. So I would love to understand that more as a teacher. But AI has been an amazing tool for us in every aspect of the procedural work we do, the logistics of getting things done.
All right, well, thank you very much.
Thank you.
Okay.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.